There we go. Now we're live. Sorry about the technical difficulties. It's been a little bit since I fired this program up, and it has decided that I am not who I say I am. Which I guess that's better than someone hacking my stuff. So my plan in this review, if anyone's even in here, I can't tell, is to just work some problems, answer any specific questions you have, or not. Yeah, like I said, I can't tell if anyone's in here, and literally have no way of knowing because everything is completely closed up for me, unfortunately. Yes, I can do some Hardy Weinberg and some phylogeny. There we go. Yep, I'll do that. I actually just go to Con and see what Con's phylogeny and Hardy Weinberg are like. Let's look. Alright. So here's Con's Hardy Weinberg. Yeah, frequently misunderstood stuff. Definitely. I can do that. Alright. Alright, so let's look at these Hardy Weinberg problems here. And go from there. So I haven't seen these or looked at them ever. So we will see what we got. Alright, so here's the first one. And some of you guys may have done these already. I don't know. It may benefit you to listen again. A group of scientists is studying a certain gene in the population of birds. This gene has two alleles, dominant F, recessive little f. The scientists collected allele frequency data for the gene twice over a 15-year period. The results are as follows. After analyzing the data, scientists determined that because the allele frequencies changed, the bird population was not in Hardy-Weinberg equilibrium, meaning that the population evolved. Which the following is the most likely reason the population deviated from Hardy-Weinberg expectations? And so this is one of which basically which one of these matches the opposite of the five things, right? The five things are the population is large, no natural selection, random mating, no gene flow, and no mutations, right? And so we have to find the opposite. Population is large, not the opposite. Natural selection is occurring, is the opposite. So we're going to go with that one. B. All right. Let's see if we can get one here. There's maybe one with some numbers in it. That's my guess is what you're looking for. All right. Group of scientists. It's always a group of scientists. Always. Dominant allele light blue and recessive is white. They found that 8 out of the 800 plants, 288 had white flowers. Assuming the population is in Hardy-Weinberg equilibrium, which of the following numbers of plants have big B, big B? Okay, good. So this is a good one. So we're given 800 total plants, and we're told that light blue is dominant and white is recessive, and we are given the number of recessive individuals in the population. Right. And so these individuals that are recessive have the genotype of. Sorry, I'm looking for my drawer here, my markers. I have the genotype of little b, right? And we know that little b is also equal to q squared, right? And I could probably type those and they'd look better, but that's okay. And so if I can just decide what's the percentage of white flowers, then I can also decide the percentage of q squared. So 288 divided by 800 is 0.36. So 0.36 is, there's 36% of the population is q squared. So that means that q is going to be the square root of that, which is going to be 0.6. That means p is 0.4 because p plus q is 1. And so if p is 0.4, then p squared, which is the same as bb, is going to be 0.4 is going to be 0.16, right? And so 0.16 is the percentage of homozygous dominant individuals in the population. There are 800 plants, so 0.16 times 800 is 128. B is the correct answer. So that was like a 2 or 3 step problem right there. Not hard, but multiple steps. It's a good question. Ooh, this one looks really complicated and really big. I'm gonna have to zoom way out to get everything in. All right, we'll see if we can pull this off. I've never had one this long before. It's because they're graphs. The answers are graphs. Oh no. It's a little bit blurry. Let's see if I can get a little bit more here. Just not going to get it all in one piece, sadly. It's just too much. So I'll get the question and then the answers separately. All right. So let's look at this. The agouti, or banded fur pattern in cats, is primarily controlled by a single gene that encodes for a signaling protein involved in hair pigmentation. Great. There's a lot of steps there. Cats with agouti fur have at least one copy of a dominant allele. Non-agouti cats. Does anybody know what agouti is? Has anybody ever heard of this? Non-agouti cats have 2 copies of the recessive allele. Number of cats with agouti and non-agouti fur, probably pronounced it wrong, in 2 separate populations shown below. If the 2 populations are each in Hardy-Weinberg equilibrium, which of the following graphs shows the correct genotypic frequencies for the population? This is lovely. Okay, so there are 400 and 400. Is that right? 400, and maybe they told me that already. And the bottom one is more than 400, actually. Looks like it's 420. Yep, okay. No, it literally is. Okay, never mind. I'm dumb. It's 200 and 210. and if they're if the populations are in Hardy-Weinberg equilibrium, which of the following graphs shows the correct genotype frequencies of the population? All right, so this is actually, so if I get a question like this, it looks like I've got like 6 things to work out. I'm going to do a quick scan of the graphs and see. What I'm basically looking to see is if the spread on each of them is different in each picture, meaning that I can just solve one of them and then find the correct picture. I don't need to solve both of them, and I think that is the case. It doesn't look like either picture is equal, or any of the pictures are, like population 2 is not the same in any of the 4 things, and so I think we're just going to solve one. So the non-fur is homozygous recessive, and that is 60 out of 200 in population 1, which is going to be 30%. And so 30, I think that's right. And so 0.3, the square root of 0.3 is 0.55. Guys, gross numbers. And so that means that Q is equal to, it just doesn't seem right. Q is equal to 0.5, P is equal to 0.45. And I feel like I'm going to have to write this stuff down. 0.55 and 0.45. And so Q squared is, I just solved for Q squared. Actually, that's probably all I need to do is 0.3 for population 1. Boom, 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 boom. It's D. I'm just going to stop. See, I was even doing more than I needed to do. I literally divided this number by 200. That was 0.3. D has a 0.3. So you don't have to solve all 6 of those numbers. You just have to solve for 1 and find the 1. Wow, and then I miss it. Okay, so I have no idea why I missed that one. That question is really busy. Honestly, I'd have to like write it down and like mess with it. Maybe someone else knows why I missed it, but I have no idea. Oh, I got it backwards. That's why I missed it. I was looking at big A, and it's actually C. There we go. I was looking at the wrong thing. I solved it correctly. I just was looking at it backwards. My bad on that. So there's some Hardy-Weinbergs. Let's see. Maybe there's one more here. We'll look at it. Yep. The graph was so small. I just looked at it backwards. All right. Population of frogs. Certain gene has 2 alleles, dominant allele and recessive. Genotype frequencies in the frog is shown in the graph, assuming the population's at Hardy-Weinberg equilibrium, which of the following is a frequency of A. Okay, so when you look at this graph, you know P squared, right? You know 2PQ. P squared is big A, 2PQ is big A, little a, and Q squared is this. And so it's asking you just A, right? And so you're going to take the square root then of 0.3, which we just literally did, and it was 0.55. Why square root of 0.3? Because this is P squared. Big A is P squared, and we're just looking for A, so that's P, and that's going to be 0.55. C. It's going to show me my summary, though. Man, that was a big celebration. All right, I don't want to do those anymore. I don't know how to use this program. Get me out of here. Okay, there we go. Yep, phylogeny. Let's do phylogeny. phylogeny definitely represents some of the more missed questions because of the way that students read the graphs and see things backwards. So, and I too will mess up, I'm sure. So we'll get to celebrate that together. All right, so here we go. That's a good problem here. The following cladogram shows evolutionary relationships among types of birds, and there they are. According to the cladogram, oyster catchers are more closely related to flamingos than to penguins. All right, so let's look at that claim real quick and just make it make sense, right? Oyster catchers are more closely related to flamingos than to penguins. Why is that? Well, to get literally think like Monopoly, right? Like if you're rolling the dice and you're moving. To get to penguins, I have to go 1, 2, 3, 4, right? From oyster catchers. To get to flamingos, it's literally 1, 2, 3, and so they're just closer, right? And that's the way you think about that. You could also say much easier, or if you can look at it, if you've seen 100 of these, you can see that they share a more recent common ancestor, right, than penguins and oyster catchers do. And so that's another way you can look at it. All right, so which of the following statements provides the best justification for this relationship? And I'll literally just walk through that. The branch of the cladogram with oyster catchers is closer to the branch with flamingos than, and I think A is good, but let's look at the other ones because sometimes I do miss things. Oyster catcher shares more recent common ancestor with flamingos. Literally, that's probably a better answer than A. Both oyster catchers are less close related to that. No, that's wrong. And oyster catchers share common ancestry, but not, and that's not true either. I think it's B. I think it's B because that has a more. Counting the things is helpful, but just seeing that common ancestor is a much better idea. Yep, good. Questions there. If you have questions, stop me too. We can look at it because since I have them copy pasted on here, we can just go back and look at it. Oh, gross. These are y'all's favorite. The ones with the pluses and everything. All right, so let's look. So here's our picture here, right? And then here's our different graph possibilities. Possibilities are endless. They're not, there's just 4. Okay. So let's look at these together. Following table shows selected derived characters for several animal species based on the information in the table, which the following cladograms best represents the evolutionary relationships among species. So plus minus indicates presence absence of the trait. Okay. The lancelet has none of the derived characters. And what did we say about the individual that has none of the derived characters? We said that's the outgroup. And the outgroup is going to be the most primitive. It's going to be the one that everybody shares a common ancestor with, right? And so the outgroup is correctly demonstrated on A, B, and C, but not on D. So we can eliminate D as a correct answer. So let's look at the one that has all the traits then. Right. Let's look at the one that has the most, or the next least, and that would be salmon, right? Salmon share vertebra with everything except the lancelet, but that's all they share, right? And so the salmon is going to be the next oldest, right? And so A shows that, and so does C, right? See how that works? But B does not show that, and so we are not going to select D or B. And so I'm just process elimination. What should go next, right? Should be newts, right? Because newts only have 2. And let's see if one of them has newt. This has newt next, and this has newt next. And so that's not going to be our eliminator. We're going to be forced to select between crocodiles and penguins. Notice crocodiles and penguins have a lot in common except feathers. And so that suggests that feathers develop later, but they're going to be very common. And then gorilla doesn't share gizzard or feathers with either one of them, but they share gizzard with each other. And so gizzard is going to come later, right? But not with gorilla. And so this one that suggests that gorilla and penguin share a common ancestor is not good, right? We would expect to see crocodile and penguin sharing a common ancestor, and then them both sharing a common ancestor with gorilla, right? And that is, I think, the correct answer. We're going to go with that. All right, so that was a process of elimination, but we got it. There might be a quicker way to do that problem, but and in reality, if I get to this problem, I can probably eliminate C or B and D really quickly, and then look at the difference of these two things here, these last two, and see that one was obviously wrong and hit A. But I wanted to build it up from the bottom for you guys. All right, let's look at this one together. All right, the following cladogram shows the evolutionary relationships among several animals. Selected derived characters are labeled on the cladogram. Based on the cladogram, which the following derived characters is shared by kangaroos and foxes, but not present in platypuses. All right, so let's look at that. Kangaroos and foxes, they're up here, right? And they have viviparity, and platypuses do not have viviparity. And that's going to be A. That one's not hard. Viviparity is live birth, if you're wondering what that was. This is another big one. A lot of these are really big. I think these that don't have names on them are actually harder than the ones that are like kangaroos and stuff, because I don't know, I can like envision a kangaroo in my mind, but I can't envision species 5. All right. So I've seen a couple of these today just because students have been working on them. And make sure you don't go to the test with in your mind high numbers, low numbers, right? Because depending on what the prompt says, those numbers can mean different things. So, yes, population genetics, I will do that. Yeah. Okay. So, in order to study evolutionary relationships, scientists compared nucleotide sequence of gene Y. So, okay, nucleotide sequence is literally like A's, T's, C's, and G's, right? 5 different species. I knew that you were wondering. I sensed that, actually, and went ahead and answered that. So, you're welcome. You're welcome. The number of nucleotide differences found between each species pair is summarized. So if there are more differences, then species are going to be less related. If there are fewer differences, they're going to be more closely related, right? And so let's look at this together. Species 5, notice species 5 here, has a ton of differences with everybody else, right? And so where are we going to put species 5? Where are we going to put it in the outgroup? And it looks like B does that, and B does is that? B is the answer. I'm not even looking at these other ones because look, this is species 4, and then I would put species 3. So it's 4, and then this is 3 and 2 together. Maybe that works. But I'm not even trying to figure out the rest of it. I don't think that. I mean, I may be off on this, but I don't think so. I think it's B. Yep. Yep. Okay. Man, celebrate me. All right. So, what population genetics? Oh, yeah. Good times. Good times. All right. In a certain population of mosquitoes, individuals born with mutation conferring resistance to an insecticide, a chemical that is used to kill insects. Because the population lives in an area where insecticides are used heavily, the mutation quickly became common in the population. A resistant mosquito from this population got trapped in a person's car and was accidentally yes, if I can find one. The half pyramid chart one. Yep. Got trapped in a person's car and accidentally transported to a different region. This mosquito then mated with an individual and local mosquito population. Over time, the mutation insecticide resistance became common in that population too. By which of the following processes did the mutation spread between mosquito populations? We're going to go with gene flow here, right? It's movement from one population to the next. And I can't click on that because it's just a picture. So we're going to go back to my screen, gene flow. Let's look at this problem together. Pollen grain from one population is blown to an area of another population where it lands on the pistil of a flower, fertilizing it. A bacterium's gene sequence changes due to DNA replication error, given the bacteria resistance to a certain antibiotic. Which of the statements best describes at least one new allele entering the population? This is actually interesting. Definitely Statement 1 does not have enough information for us to say that at least one new allele is entering the population. But statement 2, because there's been a change in the DNA, it is by definition something that is new. And so I think statement 2 alone, we're going with it. That is incorrect. Then what is it? I think it's none of the above then. No. Okay. Surely it's not 1 and 2. I think it's a bad question. Yeah. Unless I'm reading that wrong, a pollen grain from one population is blown into an area of another population where it lands on the pistil of. There's no information in that question that suggests that at least one new allele. No, that's a bad question. Not just because I missed it, but hopefully you all see my logic on that one. If I gave you that one on a test and half the students missed it, I would throw the question out. Yeah, the master is always been the apprentice. That's the secret. The secret is never stop apprenticing. That is the secret. All right, let's look at this. Between 75 and 2010, the habitat range of the island lizard population was greatly reduced as a result of human activity. The reduction of habitat range also greatly reduced the number of individuals in the lizard population. Yep, okay. Which of the following statements is most likely true about lizard population in 2010? So before I even go look at the answers, I'm going to talk about what I'm looking for. Right, I'm not even looking at them. I haven't looked at them. If you have a drastic reduction in population, what else are you going to lose? You're going to lose gene pool, which is going to reduce that population's amount of variation, its ability to adapt, and therefore its overall fitness. And so I think that I am looking for something along those lines. I think I'm going to go look for genetic diversity, which of the following statements is most likely true. Is less like 2010 population is less likely to be significantly impact? No, that's false. Lizards in 2000 are more likely to develop gene mutations. I don't know why I would believe that. 2010 populations, allele frequencies are the same. No way to know that. D is the correct answer, but I'm going to read it. 2010 population has fewer unique alleles than 1975. It's exactly what I was looking for when I came to the question. D is the correct answer. But see, I didn't even have to read it because A, B, and C were wrong. So I didn't even have to read it. I could have hit D and just went on about my business. But I didn't. All right, let's look at the next question. Scientists studying two Laysan finches in the Hawaiian Islands. Populations are isolated from each other but live in similar environmental conditions. Okay. Over a 20-year period, the scientists gather allele frequency data for certain genes, which has alleles B and B. Subset of scientists' results for the two populations show the following table. Okay. Which of the following statements is best supported by the table? So let's look at the table and let's talk about it. So we have a 20-year gap between two populations that live in similar conditions, but they're isolated, meaning that they're not reproducing with one another. And what do we see here? In population 1, we see in 95, there is a lot more big bees than little bees, right? And then in 20 years later, we see that the recessive allele is completely eliminated, which is a really strange thing because normally the recessive allele will hang on in the heterozygous individuals, but not this time. And so let's find out. So what could happen? There could have been some sort of disaster, like a genetic drift, or there could have been some sort of extreme sort of environmental changes that selected against the recessive allele, right? Let's look at population 2. Population 2 is the opposite. In population 2, you have this dramatic shift from selection for the dominant allele, which in 95, notice the populations are similarly selected. And in 2015, the recessive allele is dramatically selected for. So there's a couple of things that I'm looking for when I go down to the answers for this one. It looks like maybe my recessive alleles moved into population 2. We could maybe go find that. We could see that something selected against the recessive allele in population 1, or we could see that something selected for it in population 2. Let's go see which one of these is correct. A, allele B was lost in population 1 because of population large size. We have no idea what its size is. A is incorrect. Allele B became fixed in population 1 as a result of genetic drift. I don't think there's any way to know that, so we're going to leave that one alone. Allele big B was maintained in population 1 and population 2 because of gene flow. Nope. No, there's no way of knowing that either. So D is my correct answer here, but let's see why it's correct. Little b conferred a selective disadvantage to the finches in both populations during the study. That's not true either. So where are we going with this one? I think it's B, because I said that could have happened, genetic drift, right? And so I don't like that answer, though, but I don't think the other ones are correct. Yeah, that was right. So that I didn't like that question either, but let's read their response and see maybe a little more explanation will be helpful to you all. Let's see. According to the table, little b did become fixed in population 1 with its frequency increasing to 100% over the study period. In contrast, populations live in similar environments, so neither change in allele frequency can be definitively attributed to environmental conditions. Instead, allele V became fixed in population 1 as a result of random fluctuations in allele frequency, which is known as genetic drift. It's fine. I'm not a fan of that, but whatever. Okay. What are we looking for now? We're going to go and do... So if I do the... I don't know how con works. If I go back to the phylogeny one, am I going to get the same questions? It says I'm proficient. How do I don't even know how to do it. No, I'm not going to get the same questions. Okay. Sometimes yes, sometimes no. We'll do some more phylogeny. I think there's some concern about it. So, honestly, this unit's not too bad. Everything kind of borrows from each other, but the phylogeny is different, and Hardy-Weinberg was different. All right. The following cladogram shows an evolutionary relationship of 6 cats. Oh, look, there's the snow leopard. The domestic cat, really? Okay. Tigers more closely related to snow leopards than to jaguars. Correct, right? Because they share a more recent common ancestor. Which the following statements provides the best justification for this relationship? Literally what I just said. That's not correct. That's not correct. That D. Geckos and baboons on the same problem. But they wouldn't have thought of that in a million years. But here we are. Geckos and baboons. I can barely read that. All right. Following table shows derived characteristics set of animal things. Based on the information in the table, which of the following cladograms best represents the evolutionary relationships in species? Okay, so we get pluses and we get zeros for things that don't have it. Rays don't have anything, right? They share no common or no characteristics with anything else there, so we're going to go ahead and put them in the out group, meaning that we're going to eliminate B, which suggests that baboons are the out group. Maybe they are socially, but on this, they're not. Let's look. What's next? So geckos have just 2. Does anything else have just 2? Nope. And everything has 4 limbs except for the ray, and so the four limbs is going to be early, right? And then epidermal scales is only with the gecko, and so I'm thinking that gecko is next. And which one says that? Only one. and we're going to go with C. We're not going to go any further. All right. Following cladogram shows evolutionary relationships among several groups of animals. Select derived characters label. Literally, that's the same thing it says every time. Based on the cladogram, which the following derived characters is shared by chordates and echinoderms, but is not present in cnidarians. Chordates and echinoderms are going to share bilateral symmetry, right? They don't share notochord, they don't share trochophore, but they do share bilateral symmetry, and cnidarians don't have that. B is the correct answer. That was fast. If you have questions, let me know. But all right, here's a lovely one. I think this is the one that Ava was wanting to look at. One like this. So. All right. All these syllables up here, and that is just dog. The protein mTOR is central regulator of the cell growth and is highly conserved among vertebrates. Remember, conserved process means that everything has it. Scientists compared the amino acid sequences of mTOR from 5 different vertebrates and recorded the number of amino acid differences. Again, there's differences, so we're not looking for high numbers, or we're looking for both. But yeah, high numbers mean distantly related, low numbers mean closely related. The results of the table. Which of the following cladograms best supports the information? So let's look. This one's interesting. So one of the things that I've noticed is I've seen several of these problems as you guys have showed them to me. These tables really are trying to like make you think and look, right? And so think in groups, right? So what groups do I see here? Well, I see some 200s, right? I see some 200 numbers. I see some 100 numbers, right? Now this isn't foolproof, but this is just kind of the way that I'm looking at this problem as I look at it. And then I see that 4. Right, and so where are all of those 200 numbers? They have to do with zebrafish, right? So zebrafish is going to have the most differences, okay? And so it's going to be the outgroup. B has it as the outgroup. A does not. B has it as an outgroup, and D does. So we're deciding between B and D. And so then look at the hundreds, the green numbers. They all have to do with emperor penguin, right? Here and those two. So emperor penguin is going to be next. Which one shows that? Both of them do. So that is we're not going to be able to eliminate using that. What's next? Dog has 15, right? So dog is going to be next. Which one shows that? Not B. B shows baboon. And so here we have dog being the next, then human and baboon sharing a more recent common ancestor because they only have 4 differences. So D is the correct answer. Another way you could do that is you could see, and you could work the other way, and it's probably fixed to where it will take you the pretty similar amount of time. And it's honestly what I just did could take you about, 12, 15 seconds if you were not speaking it out loud. You were just doing it. So, but yeah, I don't remember which answer I said. D. Okay. So yeah, I feel pretty good about those. I don't know where you guys are at on that, but I don't feel like you're going to see anything that's more difficult, or I don't remember what's on the test, frankly, but let's see. It's about 9 o'clock, so it's getting pretty late. Is there anything specific that we want to go over before I get off here? Obviously, if you guys have questions, you can come in the room in the morning, and I'll be there. Apparently, there's an AP Chem test tomorrow, too. Just lovely. Yeah, there's a lot of reading graphs in this class, actually. You can't come to the graphs without content knowledge, though. You have to have some content knowledge for the graphs, but it is a lot of reading graphs, reading pictures and graphs and things. Yeah. I'm not going to say this is an easy unit, but I will say that test averages are significantly higher than the other units. Doesn't mean like lay down on it or anything, but I think that you guys will be fine on this test. I'm not that concerned. All right. Well, it has been fun. Hopefully this is helpful. If you have questions in the morning, come on in and we'll work through it. My guess is that I'm going to have a lot of freshmen in there working on Punnett squares tomorrow too, because my lands, it's pretty rough. I mean, you could probably pass it. I think you could pass it. In that the passing is, a 60 or above. I think you could rock it out. I think you could do better than pass it. It's actually not a difficult exam. This is material you guys have had a bunch of times. So, all right, it has been fun. Y'all have a good night. Do not stay up too late.